Students will often ask how can they be successful on their practical exams or what is the best way to study. While there are several ways to study, I do have some retention and success strategies for your practical exams that will follow through these steps. You can also find this document in your Canvas, and then we'll also walk through where to find each of these things that are mentioned in this document. So, the first thing to note is that it is important to note that you are going to be learning lots and lots and lots of structures. And so, therefore, actually just trying to commit them to memory by themselves more than likely is not going to lead to success because of the sheer factor of how many things you will be looking at. Therefore, you need to work through how you can go and get to a recall phase so that you can recall the structure during the exam or during the practical. The steps below are going to be a good start at what we call space practice. Space practice allows you to use repetition spaced out over time of doing almost the same thing, but adding another layer to it that commits it to your recall. All right, so the first thing that is going to be noted here, you must utilize, it is required to use the textbook from the lecture course and the lab manual. Without the two of these resources, your success in this course will more than likely not occur. So, with that being said, if you are not enrolled in the lecture for this course, please send me a message in Canvas with your name, your course and section, and your email letting me know that you're not enrolled in enrolled in lecture. All right, so the textbook and the lab manual. How do you find those? Well, in your Canvas course here, you will click on Wiley Course Resources on the left-hand side. It'll bring you to a page that will have these squares, course resources, practice material. Also goes through each exercise here. Each exercise here has other either animations, worksheets, concepts, things like that can help you. But right now, we're going to get to this textbook. So the first square is the e-textbook. You'll click to launch it, and that will take you over to our textbook. Here for your textbook, you will then find, it'll take you to the place you left off. And on the left menu, you could just change to where you want to be in the table of contents. So this is your textbook. I'm actually going to click into exercise 2 because it has a little bit more. Well, actually, we'll stay in exercise 1 for the beginning. So you have an introduction. Most of this talks about the physiology of things. Right now, our focus is going to be on anatomy and the structures. So after the physiology portion in the lab manual, you'll then find structures for the practical. It is called gross anatomy because gross meaning large here, anatomical structures. And so it will tell you on the models, find the following structures and organs. This is your checklist. This is your study guide. This is your tool to know what it is that you are expected to know and be able to find for the practical. And so therefore you will use this as a checklist, and you'll see how I recommend doing that in the next set of things. Also, if the exercise has histology is the study of tissues. So it's where we look at the cells and the tissues that make up these structures. And so this first exercise, the endocrine system, does have histology. You will then, after a little bit more about reading and seeing what it does, you will then find a checklist again, and hence it says checklist, of what you need to be able to identify on each of the slides. So from the pituitary gland, On scanning power, you need to be able to identify the adenohypophyses and the neurohypophyses. And then when you get to the thyroid gland, you need to be able to identify the thyroid gland and the follicles on scanning power, as well as the parathyroid gland. But then when you move to a higher power, you would need to also be able to identify specific cells and colloid. So this is a great checklist for you to use, and it is going to be how we also can work through the steps that I recommend for success in the course. Also, if you are enrolled in the lecture and you have access to the lecture textbook, remember, even if you're not enrolled, you're going to let me know. And you will then click into this Wiley Reader bookshelf if you want to move or navigate between the lab manual and the textbook from the lecture. You'll click that, and then you can find your actual lecture textbook here. And so you just navigate between the two back and forth in that method. You're going to need this textbook. We'll go back over to the endocrine system here as well. You'll need this textbook because you are going to depend on the labeled structures in the textbook to also help you identify the structures you are labeling or your models that you're labeling. So let's head on back over to the tips. All right, so now that you've gotten both of those, textbook and lab manual, and you can find them, you know how to use them, you know what you're looking for in them, the next thing you're going to need also is going to be at the image gallery. You have an image gallery in every single module of your course. So, for example, here in exercise 1, under the virtual lab resources, you have exercise 1, endocrine model, image gallery. It has a little note here that you should label all of them for the practical, because that is how we recommend that you be successful in this course. And then below that, you'll also find some additional things that are helping. Most of the time, you'll find that we always have videos that we provide. And so the model videos and the histology videos will be here. Additionally, sometimes you may find some other things. So for example, in exercise 2, the coronary blood vessels that are in your textbook are not as extensively labeled as we are asking you to find. And so you have an actual PDF document of a printed structures that are labeled so that you can utilize those. So you may also sometimes find that because you're also looking at some sheep hearts. We have dissection videos on the heart, and we also have a dissection walkthrough. So other things will be there. So now this image gallery. I recommend that you print it, but if you are not a print person, if you are a digital person, then there are options for that as well. So these models here are in PowerPoint, or these image galleries are in PowerPoint. If you want to be able to convert them to a PDF, of course you can convert your PowerPoint to PDF, or if you want to take your PowerPoint and just write on it directly through PowerPoint, adding labels and lines, you can also do that. We provide two to you. One is the models only, and one is going to be the models with the corresponding textbook images. So this is a good start. This is where you should start. The ones without the textbook image will be your progression, that space practice that we're getting in. So of course here, it tells you which models you're going to be using and finding. This won't be like this if you actually open it in PowerPoint. This is a conversion thing with Canvas and Chrome. But you'll notice here you have the first model. Next to the model, you have the textbook figure 18.1. And so you are then able to label these based on, so you have your textbook image, so you need to then pull up your checklist, right? And so then you are able to take this checklist and you are able to go through. It actually tells you those same things in the checklist here, but you are able to label those and go through. We do the same thing here for the models. And then also in this image gallery, you will find histology. And of course, what should you be able to label on this? So that checklist again that we mentioned. So when we get to the thyroid here, scanning power, what you should be able to find. So this checklist is actually provided for you here in addition to in the textbook or in the lab manual. All right, so first time through, use those images next to the model and label the things on the model. Also, as I mentioned, you do have videos. The videos that you have will walk you through pretty much going right along with that checklist in the book, and they'll show it to you on each of the models. They are, for the most part, in shorter little segments. So for example, here with the heart model videos, because the endocrine doesn't have a lot of structures, that's probably the smallest number of structures you're labeling all semester. With the heart, for example, you'll see that you have multiple videos as they're trying to load so that you can get each part of the heart. And this makes it great if you find that you're struggling with just the coronary circulation versus the external heart anatomy. You don't need to watch this video over and over again or have to skip through to get to the part that you actually need. They're sectioned off for you. So first thing to do is make sure you label the images using the textbook images and the model videos. That's your first time through. That's what I recommend. And then your goal is eventually to work up to where you are labeling those images without any kind of word bank. The lab manual would be considered your word bank, that checklist, right? Because you're not going to have that on your exam. And so you want to work yourself up to that. And so maybe after you've labeled them one time through with using the videos and the textbook images, the next time you label them, and instead of using the images with them, and instead of using the videos, you do utilize the lab manual checklist though, but you utilize that as a word bank to find the structures you need. And then maybe the third time through, you don't utilize anything, you're just trying to label. And so, of course, this will be good. In the midst of doing all of this labeling, you would then want to complete your pre-lab, right? So generally, I say, your pre-lab, yes, you can look the things up, you can find what it's asking you, and these quizzes and pre-labs and post-labs and all of the other practice things that you have found in your modules to help you label anatomically, like the practice practical and that, all of those are great, but they do not allow you to practice labeling all of the structures which you may be quizzed on. And so therefore you need to make sure you label and then utilize these videos, utilize these practice quizzes as an actual quiz, not to look up what it's asking you, because then you won't be able to perform well in your exam because you're not going to be able to look up what we're asking you in the exam. So make sure you go through and label. The other good thing about the labeling is, especially when we start moving into the heart, the digestive system, the blood vessels, most of your body is a flow from one area to the next. So for example, the same way you would give someone directions to get somewhere, right? You give them the directions in the order of which they're going to pass the places and make the turns. You don't give them the directions out of order. You would not study these structures out of order, because therefore you won't really learn them or get there. And so just remember, there is much, much, much benefit to making sure that you label these structures with the lab manual and checklist. If you have any questions, please don't hesitate to contact me. I'm available via Canvas message, via email. If you message via Canvas or email, please allow 24 hours during the week for a response if it is a weekday and not a holiday. And if it is the weekend, please allow to the next business day. Also, office hours do exist. You can come ask a question. You can go over some things. We can go over past exams. If you're having a problem, we can definitely discuss during office hours. And then finally, Utilize the resources that are available to you in addition to myself for free that are from Delgado for tutoring. We have a Science and Math Resource Center, and then there are also online tutoring options that are very, very helpful. If you have questions, like I said, please reach out, and good luck this semester.